Hello, my name is Robin Nusslock and I'm from Northwestern University. I'd like to thank Dr. Martha Farah for asking me to speak in this symposium titled Finances and Feelings, the Affective Neuroscience of SES. I'd also like to give a special thanks to the Program and Planning Committee of CNS. I wish I was speaking to you in person, but CNS has done an extraordinary job putting together this virtual meeting. The goal of my talk is to examine the role of brain systems involved in both emotion generation and emotion regulation in helping protect people from the mental and physical health consequences of poverty. In short, poverty hurts. Low socioeconomic status is associated with both mental and physical health problems across the lifespan, including asthma and respiratory infection, in childhood, depression and substance use, in adolescence, and metabolic syndromes, coronary heart disease, and psychiatric disorders in adulthood. Now, researchers have done foundational work examining the correlates, causes, and consequences of poverty on the brain. And this has really spearheaded a new field examining the neuroscience of socioeconomic status. Extending this work, we'd like to suggest that a multi-organ perspective may contribute to our understanding of how stress, including poverty, gets underneath the skin. A particularly good candidate for such a multi-organ perspective is the immune system. The brain and the peripheral immune system are in constant communication with each other. The brain can modulate immune signaling in the periphery, and this peripheral inflammation can access the brain and affect emotion, mood, cognition, and behavior. And so for the present talk, I have two goals. First is I'd like to provide a neuroimmune network model as a conceptual framework of how poverty and life adversity generates risk for emotional and physical health problems. And second is I'd like to examine the role of certain brain systems involved in both emotion generation and regulation in protecting people from the health consequences of poverty. This is the theoretical foundation for this talk is the paper that I recently published with my colleague Gregory Miller. And the core question of this paper was addressing how is it that early life adversity, and particularly poverty, get underneath the skin to generate risk for both mental and physical health problems. And relevant to this research is considerable evidence that early life adversity, such as poverty, can sensitize cells in the amygdala to be very sensitive and very responsive and very reactive to threatening stimuli in the environment. And this is some of the research demonstrating this. We know the amygdala is involved in threat processing, vigilance, and various forms of anxiety conditions. And there's evidence that early adversity, including poverty, is associated with structural and functional alterations to the amygdala in a manner that make it more responsive to threat, and that these effects of poverty and stress on the amygdala sustain throughout adulthood. In a paraliterature, there's evidence that early adversity also sensitizes cells in the immune system that are involved in generating and facilitating inflammation. Inflammation is one of the first responses of the immune system to irritation or infection. It's stimulated by chemical factors, including pro-inflammatory cytokines, that are designed to facilitate tissue repair and the clearance of pathogens. And when regulated, this is a highly adaptive system. We would die without it. But when dysregulated, and when it results in chronic inflammation, you can have a host of mental and physical health problems that emerge as a result of chronic inflammation. And in fact, these are very similar health problems to the problems that are associated with early adversity and poverty. So inflammation may be a mechanism through which stress generates risk for mental and physical health problems. Just to give you an example, this is a study in which they looked at children, 267 children between the ages of 8 and 10, and they drew metrics of inflammation via the blood. We're looking at the y-axis. This is interleukin 6 or IL-6, which is a pro-inflammatory cytokine. On the x-axis, we have socioeconomic status, and you can see that individuals with low SES, i.e. individuals living in poverty, have increased inflammation relative to individuals who are not living in poverty. Now, the mechanisms through which early adversity get underneath the skin to create this pro-inflammatory phenotype appear to occur in part through the amygdala. And in particular, under conditions of stress, the amygdala will activate a biological cascade, including the activation of the sympathetic nervous system and the AHP axis. The sympathetic nervous system innervates organs where white blood cells, and these classical monocytes, develop, including the bone marrow, the thymus, and the spleen. And it releases norepinephrine onto these early immune cells and can increase inflammation. And this is highly adaptive. When the brain is in a state of threat, it wants the immune system in the body to be in a state of threat and well. And again, when regulated, this is highly adaptive. But when dysregulated and under conditions of chronic threat, you can have conditions of chronic inflammation. Now, it appears that the signaling between the brain and the immune system goes in both directions, from the brain to the immune system, as well as from the immune system to the brain. And there's compelling evidence that pro-inflammatory cytokines can get into the brain and modulate brain areas that we care about as cognitive and affective neuroscientists. So pro-inflammatory cytokines can access the brain through leaky portions of the blood-brain barrier. It can enter at circumventricular organs. It can initiate active transport of endothelial cells in the brain. And one of the primary areas in which the immune system in the body targets in the brain when it accesses the brain is the amygdala, because when the body's in a state of defense, it wants the brain to be in a state of defense as well. So increased inflammation in the body has been shown to increase amygdala signaling to threat in the brain. Another important target for inflammation in the body when it accesses the brain are reward systems in the basal ganglia, including the ventral striatum and the nucleus accumbens. So what has been demonstrated is that if you have increased inflammation in the body and when this accesses the brain, it can lower or attenuate or reduce reward-related brain function in regions such as the ventral striatum. Again, this is highly adaptive because what it's doing is it's inducing a set of sickness behaviors to slow down goal pursuit and approach-related behaviors and preventing people from getting up off the couch and going out and pursuing rewards, and instead to sit still and rest and recuperate. This is, again, highly adaptive in the short term, but under conditions of chronic inflammation, this can generate chronic reductions in reward-related brain function, which we know are associated with conditions like depression. Just to give you a proof of concept, Naomi Eisenberger and colleagues did a study in which they injected individuals with an endotoxin, lipopolysaccharide, which is a molecule on the surface of bacteria. And after injecting this molecule into the person, this creates a pro-inflammatory environment in the body. They exposed people to a monetary incentive delay task, a reward paradigm. And what you can see is on the right figure, the endotoxin group, which is the group that got the increased inflammation, shows reduced reward-related brain function in the ventral striatum relative to individuals who got a placebo, providing proof of concept that increased inflammation in the body can reduce reward-related brain function in the brain. You also can see in this study in which they did PET imaging that this appears to actually involve the reduction of dopamine signaling. So in particular, inflammation appears to reduce dopamine signaling in the ventral striatum, creating a profile of low positive emotion and low reward-related brain function. Finally, there's evidence that peripheral inflammation also can modulate the structure and function of the prefrontal cortex. And so what we have here is we have inflammation that's actually creating a neural phenotype in the brain that looks very similar to what we see in forms of depression and various forms of psychopathology, including high threat neural activity, decreased reward-related brain function, and lower prefrontal executive control. And this is a meta-analysis in American Journal of Psychiatry just showing me that depression is associated with heightened amygdala activation, decreased reward-related brain function, in this case in the caudate, and decreased dorsolateral prefrontal cortical activation. Again, suggesting that inflammation can create a profile in the brain that looks like low positive emotion, high negative emotion, i.e. depression. Just finally wrapping up this conceptual model, then what we propose is that when the brain is in a state of dysphoria, when the person is in a state of dysphoria, they will then go out in their environment and engage in unhealthy, high-risk behaviors in order to assuage their negative emotions. And a lot of these high-risk behaviors happen to be pro-inflammatory, like high-fat diets and smoking. And so this can create a positive feedback loop between a brain that's characterized by negative emotions and behaviors that further increase inflammation. That inflammation, when it cycles back to the brain, can further enhance the neural phenotype associated with depression, and over time this can spiral out of control, leading to a state of neural activation characterized by depression and increased inflammation. And so with that model of how adversity, including poverty, gets underneath the skin to generate risk for both mental and physical health problems, I next want to present a series of studies examining the possibility of both emotion generation regions and emotion regulation regions in protecting people from the health consequences of life adversity, and in particular of poverty. And the kind of core question here is that if we return to this figure that I showed earlier of showing a relationship between socioeconomic status and inflammation, you can see that the mean levels of inflammation are higher for the low SES group, but there's also substantial variability. And so what we want to do here is to say, well, what brain systems, and particularly what brain systems in the emotional brain, might protect people from the effect of poverty on both mental and physical health. So the first study that we are looking at for this portion of the talk is asking the question of whether or not threat reactivity in the amygdala might actually help explain why people have particularly profound responses to poverty in terms of their physical health. And for this study, we scanned 250 youth from urban Chicago. 57% of these individuals resided in low-income households, giving us a good variability of socioeconomic status. We then had participants complete a phases paradigm to assess threat activity, and we focused on activity in the amygdala. And we then grouped participants based on their reactivity in the amygdala during the phases paradigm. And we, in particular, grouped participants to be high reactive in the amygdala, average reactivity in the amygdala, and lower reactivity in the amygdala. And what you see here is that on the x-axis, we have household income to poverty, where lower numbers here reflect higher levels of poverty. And on the y-axis, we have low-grade inflammation, where higher numbers reflect increased inflammation. And what you can see is that at high levels of socioeconomic status, there's no relationship between amygdala, there's no difference between levels of inflammation depending on people's amygdala reactivity. But at poverty levels of socioeconomic status, so at the far end of the x-axis, what you can see is that individuals who have low reactivity in the amygdala are actually pretty unaffected by poverty. But if you have high levels of threat reactivity in the amygdala to threatening stimuli, you show increased levels of inflammation, suggesting that the amygdala moderates the relationship between poverty and inflammation. Now, a question that we then ask, well, what drives this reactivity in the amygdala? Is it inherent in the amygdala, or might it involve top-down regulatory control of the amygdala by prefrontal brain systems? And to address this question, we're going to focus on the central executive network. And the central executive network was studied in this particular study using resting state functional connectivity. And so we're interested in the extent to which emotion regulation can help protect people from the negative effects of poverty. The foundation for resting state functional connectivity is the discovery of this functional architecture in the brain when a person is not engaged in a task. And the advantages of resting state functional connectivity are a few. One, it allows you to examine the brain when it's not during a task. And also there's evidence that particularly when you get high numbers or a lot of resting state functional connectivity, you get fairly stable metrics of trait levels of profile in the brain. And so this has its advantages. We particularly focused on the frontal parietal central executive network. The frontal parietal central executive network is implicated in cognitive control and executive control. It's implicated in emotion regulation. So it's a network of the brain that's involved in regulating attention and emotion, and it's anchored in the dorsolateral prefrontal cortex as well as the posterior parietal cortex. In this particular study, what we were interested in looking at was the extent to which central executive network might actually protect people from some of the physical health consequences of living in poverty. And high rates of poverty are associated with high rates of neighborhood violence. So there's a covariation between poverty and neighborhood violence. And youth who are exposed to high rates of neighborhood violence display cardiometabolic risk factors that include obesity, high blood pressure, allostatic load, etc. And these cardiometabolic risk factors in children have been shown to increase risk for coronary heart problems as adults. And importantly, cardiometabolic diseases appear to be mediated in part by increased inflammatory signaling. So high inflammation appears to be a pathway through which stress gets under the skin to generate risk for various coronary heart problems. What we predicted here was that high central executive network connectivity would protect people from the cardiometabolic consequences of living in a violent neighborhood, and also as a corollary, living in poverty. So would high central executive network connectivity protect people from the health consequences of living in poverty? We studied 218 urban youth in this particular study, ages 12 to 14, and they completed a resting state scan. This is a map, a heat map of the participants in our study. So the black dots on this screen here reflect each participant. So each participant is a little black dot on this heat map. What we then did is we did a coding system in which we plotted the number of murders in the city of Chicago for the 5 years prior to a person's fMRI scan. And hot colors on this heat map reflect high numbers of murders, and the murder rate for these hot portions of this heat map in Chicago are much higher than the average for the United States, whereas cool colors such as blue reflect low rates of murder in the city of Chicago. And then what we could do is we could examine the relationship between where somebody lived in their exposure to murder and the health outcomes as moderated by central executive network functional connectivity. What you see here is on the x-axis, we have neighborhood murder rates, where higher numbers reflect higher murder rates and as a corollary, lower socioeconomic status. And on the y-axis, we have a metric of insulin resistance, which is an important variable in cardiometabolic risk. What you can see here is that for people who are living in a high murder environment or a low socioeconomic status environment, they have very high levels of insulin resistance, which is, again, a risk factor for cardiometabolic health conditions if they have low resting state functional connectivity in the central executive network. By contrast, for people who are living in a high murder environment or low socioeconomic status environment, people with higher resting state functional connectivity in the central executive network were essentially fully protected from the health consequences of poverty. And we see no relationship between the central executive network and cardiometabolic risks for participants living in a low violence neighborhood. And we see this across multiple indexes or indices of cardiometabolic risks. So what this is saying is that if you're living in poverty or in a high violent neighborhood and you are have a strong central executive network, you may be engaging prefrontal brain systems and regulatory systems that are quieting subcortical threat systems in a way that are protecting you from the negative health consequences associated with living in poverty. Just as an extra point, we found no relationship for other standard networks of resting state functional connectivity, including the salience network or the DMN, and this adjusted for a number of demographics. So if a high resting state functional connectivity, or to put differently, strong prefrontal regulatory strength helps protect individuals from the negative health consequences of poverty, a follow-up question to that would be, well, how can we actually promote strong prefrontal regulatory strength amongst people who are living in adversity? And there's compelling evidence that supportive parenting seems to be a major protective factor for the negative consequences of poverty on both mental and physical health. And this is largely the work of Gene Brody. And so what Gene and Brody and others have shown is that parenting that involves a high sensitivity, high emotional support, low conflict helps protect people, and particularly children, from the consequences of poverty. And so what we did in this study is we examined 119 African-American young adults living in rural Georgia. They completed a resting state functional connectivity scan. And we looked at the relationship between resting state functional connectivity and years living in poverty as moderated by the degree of supportive parenting that they have by their parents. And what you can see here is that amongst children who are living in poverty and who have a low supportive parenting style, or whose parents have a low supportive parenting style, you see the expected relationship between years spent in poverty and the central executive network. In particular, people who are living in poverty have decreased central executive network functional connectivity, i.e. decreased prefrontal regulatory strength. But people who live in high supportive parenting families, children whose parents are very supportive and are high on this index of supportive parenting, are again fully protected from the deleterious consequences of poverty on the central executive network, suggesting that supportive parenting may create a neural profile of resiliency in children that may in turn protect them from the negative consequences of poverty. So just to provide a synopsis of my talk, what I first did is I provided a neuroimmune model that helps us better understand how poverty gets underneath the skin to affect both mental and physical health. And we suggest this is a two-hit vulnerability for the health consequences of poverty, both the mental and physical health consequences of poverty, such that poverty creates a neural profile associated with depression. It can create a pro-inflammatory phenotype in the periphery of the body. When chronic, those two systems can become dysregulated and amplify each other and generate a prolonged risk for both mental and physical health consequences. We showed that people with high threat reactivity in the amygdala appear to be particularly at risk for the health consequences, or the negative health consequences of poverty. And by contrast, people with high functional connectivity and emotion regulation and executive control networks appear to be at lower risk for the health consequences of poverty, suggesting that strong prefrontal regulatory strength may be a neural marker of resiliency for the deleterious consequences of poverty. And finally, we suggest that supportive parenting can strengthen resiliency of poverty, and it may do this in part through strengthening the emotion regulation and executive control networks in the brain. So with that, I would just like to thank my colleagues, Greg Miller, Anita Chen, and Jean Brody in particular, for collaborating on this work. And I'd like to thank CNS for hosting this virtual meeting, and thank you for watching this talk. Thank you so much.